Thomas, it's a great pleasure to welcome you here. Thank you very much for spending some time with us to record this discussion. You've had a long, successful career and made amazing contributions to basic science understanding oncogenes and how cells can be transformed, and it would be great to hear a little bit of your perspective on how your field has changed, what you've achieved, but also the difference you've seen as the field has evolved over the last few decades.
You know, my main focus at EMBL was, as you say, on oncogenes that were actually cellular genes that were carried by retroviruses and that would convert normal cells into an abnormal behavior which made them cancerous. And that was both growth control, but also often changes in their makeup as specialized cells. So they acquired different phenotypes. And this connection between the oncogenes and the cell differentiation, specialization of cells has always fascinated me. And so this eventually carried me over to try to understand, you know, how cells decide what to become. And here, of course, transcription factors have a fundamental role, and many of the oncogenes that we and others described for the first time turn out to be transcription factors. And so we basically followed work that we started here and before at the DKFZ into a field which is much more related to cell fate decisions, but the thinking is very similar actually, because, you know, in the end, you know, transcription factors are key molecules that switch genes on and off, and so they do this in the context of cancer, they do this in the context of differentiation, and in the end it boils down to trying to understand, you know, the complex interactions between them and the many levels of at which they work. But essentially it's a almost continuum of what I've started here. Oncogene cooperation, for example, we see this very often now in differentiation, transcription factor cooperation, or situations where they actually are enemies, where they fight each other, and so the concepts are not so different. But of course, the tools have tremendously improved, and the insights that we have are amazing. And of course, unfortunately, the more we understand, the more complex things seem to turn out to be. You know, there are still. It's hard to say where we are really in understanding cell fate. I have no idea if we would be, you know, 20 years from now, that we would say, well, we have already understood 80% of what's important. I have no idea. It's hard to say. I think there's a lot more to learn.
Yes, and you brought up a very important point, how technology affects what you can know, what you can measure, and we've seen over the last few decades remarkable advances when you know, when we think back to the time when we were in EMBL, the idea of just being able to sequence easily sequence and inexpensively sequence every genome you want to, and also measure proteins. I mean, this is something I became very excited about, how technology breakthroughs transformed identifying and quantifying proteins from a dark art that was super difficult to something now we can do at huge scale. Have you seen that impact of high throughput technologies in your field?
Absolutely. I mean, it's absolutely fundamental. You know, everything we do now is based on some kind of omics technology, and lately, of course, more single cell analysis as well. That there's so much to be learned very quickly by applying these omics approaches to the questions you're interested in, which is sometimes also daring, because in the end, what is almost rate limiting is clever bioinformatic analysis, and there's so many ways of doing it, and I think this is now almost the bottleneck, the really applying, and there's also continuously developing of new tools that you can use to analyze your omics data. And I've been in a lucky situation that I've collaborated with very good bioinformaticians, and that makes a huge difference.
Yeah, I think that's absolutely right, Thomas, that we now live in an era of big data. And so we have to become, at some level, computationally literate and computational biologists and embrace the opportunity it brings. But I often comment to colleagues and people in the lab that nonetheless we shouldn't mistake data for knowledge. And I think one of the big challenges in all these fields is the ease of now recording large volumes of data doesn't mean it's easy to turn that into really important insights that advance the field. And that's one of the exciting challenges ahead. I'm sure that's going to be true also in your field.
Yeah. I've always said to my people, don't go too quickly into the molecular biology of something that you're studying into this computational analysis and so on. Unless you're really convinced you have a biologically interesting question that you are dealing with, and that this is important for the biology which is behind it. Otherwise, you waste your time. It's much easier to go into this using these tools and cranking out data than really identifying a new interesting angle of the biology. And so I think of course the biological understanding of what you're doing is going to be fundamental also for the future. It doesn't help you to, as you say, to generate huge amounts of data that are perhaps mostly descriptive without really seeing the connection to the biology that's behind.
Maybe necessary but not sufficient.
Yeah,
Would
absolutely.
be a way of saying.
Absolutely.
And we still don't have reliable cures, sadly, for many forms of cancer, which I know is a huge interest of yours. And despite the huge amount of time and effort that's been spent, how much do you think this reflects a lack of basic knowledge still about the underlying mechanisms and interactions to make druggable targets accessible for new therapeutics? Is it still a lack of basic knowledge, or is there some other major problem that's preventing progress in clinical therapy?
Yeah, cancer is such a large collection of different diseases, and I think for some the basic knowledge is probably, and we know it, that there are some cures based on tyrosine kinase inhibitors, on research in the lab, and it has led to very important advances in cures. But then there are other types of cancer that are evolving so fast, continuously mutating, and it's sort of a moving target. And so you develop one line of therapy. I mean, that's at least my impression. Let's say a novel treatment based on a compound, but then in relatively short time you will find new variants that escape. It's a little bit like with COVID now.
Exactly. I was just thinking maybe as we find some of our colleagues unable to attend today because of yet another wave of new COVID variants that are infecting, is this just simply unavoidable with viral infections?
Well, I mean, COVID I think is a very unique virus. I mean, I don't think we have seen anything like it before. And I mean, if you want to talk about COVID, from a virus point of view, when I saw the news coming out of China that this virus has a latency period in which it is already infectious but causes no symptoms, I became extremely worried that this could be terrible for as a pandemic. And I've actually alarmed people in our director, we should think about how to react in the institute at that level. At that time, they thought I was a little bit overreacting. But this is of course a terrible situation. With flu, you're basically only infectious when you have the symptoms already, and so it's very hard to do anything about virus spreading. And of course, there is no. I don't think there is a strategy right now that people think agree on that is the best one, and this is a little bit like with cancer, that we are learning a huge amount, and we are coming out with all kinds of therapies. I'm sure that actually eventually we will be able to have the capacity to treat most forms of cancer with highly specialized, personalized treatments, like which are being developed by the people who developed the BioNTech vaccine.
So our personalized medicine, precision.
So they're basically charting all mutants in the tumors and then targeting with a specific RNA.
But I think
vaccines.
This reflects the very topical point of diversity, which is a word we hear a lot about in our society in many different directions, but diversity underlies genetics and evolution. And I think, what you're saying with cancer, you see and with viruses like COVID, you see evolution in action. You see the arms race where rare variants arise by chance, but are then selected powerfully by their ability to fit, to be very well adapted to a particular opportunity in the ecosystem they're living in. And perhaps we need some more understanding of how to design therapeutics to take into account this inevitable arms race of host pathogen interactions and the constant throwing up of genetic variation.
I mean, this of course has been practiced a long time, for many years with influenza, where labs are developing vaccines expecting the next big outbreak, and sometimes these vaccines are actually work quite well and are useful, but sometimes I think like the last one, they're completely off. So the vaccines that have been developed are basically useless, and I think it's going to be very difficult. There's so many possibilities of mutations, even in a single protein like the envelope protein of the coronavirus, that I don't think it's going to be possible to develop algorithms which can safely predict what's going to be the next big one. My impression, but I might be wrong. With artificial intelligence, maybe it can be done. I don't know.
Well, maybe. Artificial intelligence is, of course, another exciting, rapidly developing field that creates new opportunities. But since we don't know if this will be useful, we certainly shouldn't make our plans relying upon it. But I think developing rapid response capabilities is probably a more wise approach. I mean, the coronavirus pandemic was shocking, a big element of change in society, and it also revealed a lot of things about how unprepared we were as a society. And also, I think...
Yes and no. Yes and no. I mean, it's unbelievable how fast these vaccines were produced. So we were prepared not for a virus pandemic, but the tools were developed actually for cancer. And so, luckily they were fully prepared to immediately apply them.
I would say it slightly differently. I'd say that we weren't prepared, but because we had a strong basic science infrastructure, we were able to adapt to refocus, to do it. And then the concern for me is that it was very fortunate that basic science came to the rescue, and the new tools that had been developed could be adapted to make with unprecedented speed.
No, I to continue what you were saying, I think if this pandemic would have caught us 5 years earlier, there would have been hundreds of millions, well, hundreds, I don't know, but certainly a lot more people that would have died. These vaccines were absolutely essential to keep us out of the worst.
Yes. Thomas, you've had an amazing career that has moved across continents. I know you've lived and worked in Europe, in North America. I believe you lived in South America as well. And you've been a world-leading scientist in institutes also in Germany, in the USA, and now in Spain, in Barcelona. What are your perspectives about performing science and how science is carried out in these different parts of the world? Is it all the same, or do you notice distinct differences?
One of the first things I noticed when I started as a professor at Albert Einstein in New York, that people were talking about other scientists' achievements, not so much in terms of science, but how much grant money they got. Money was absolutely always sort of almost the top.
The metric of achievement.
For everything. And so science is very much dominated by how much you're capable of attracting money, and that creates a different way of how you do science. You know, I was very impressed by some of my colleagues who were incredibly good at getting grants, even though they were not such great scientists. And I was struggling in getting my grants because I didn't have these abilities that we were not used to. We were not trained to do this here. So there was a big difference. But on the other hand, I must say, I also was very impressed by, Americans are also very good at communicating and making collaborations across many places in the country and worldwide, and reacting very fast to new developments, much faster than many of my European colleagues. What I noticed is that even though many institutes here had hardcore funding for the researchers so that they didn't have to struggle so much for grants, the Europeans were doing more of the projects that were already sort of the threatened paths. They were not so innovative. The Americans were
maybe more in a comfort
innovative
zone.
Yeah. And even though they were in much more risky positions, because their own salary was dependent on grants and so on, they were doing more risky research, and that I thought was very inspiring. Our institute in Barcelona is very different from the way research is done in typical institutes in universities in Spain. We were modeled completely along the EMBL model. So we have the same structure with contracts that only last 5 years for group leaders, which can be extended maximally to 9 years. And that makes a huge difference. And of course, we also became very international, which was untypical for Spanish places. And so I think we managed to actually bring this model into Spain as maybe the first institutes. And now there are more institutes that are doing this. Creates more mobility. And I think this is a great example of EMBL having a huge impact in the way research is done in Europe. And we are not the only institute, of course, that is modeled along this.
Yes, but I agree. I think that's one of the great ways EMBL has and can continue to deliver impact, by setting some examples from the top down of what are the real things to value. If we look forward now into the future, it's hard to look very far forward when things change so fast, but if we just think over the next 5 or 10 years, where do you see the next really exciting breakthroughs coming? In fact, you mentioned earlier the development of tyrosine kinase inhibitors as cancer therapeutics. I think a lot of people predicted that it would not be possible to develop specific inhibitors to kinases because there are so many kinases with similar structures. And I think that also, for me, highlights that we shouldn't assume we know too much, and you should let people still try things that the experts think maybe that won't work. Because otherwise, you just don't have these surprising breakthroughs. Yeah. If we become too conservative and always look for why things might not work, we have to encourage curiosity-driven, crazy ideas.
Yeah, I mean, that brings up a thought, when I talk to students, especially when they start in my group, I always say, first, never take for granted that what's published or what I say is correct. Always, try to challenge me if you can. And if you propose an experiment where I tell you that I disagree, I would encourage you, at least in some situations, do it anyway, because I might be wrong. Yeah. And so, I think it's important that people have their own initiative, and even though probably in 9 out of 10 cases it's wrong, the one experiment that works is
pays for all.
Pays for all.
No, I completely agree, and I think that's an important message that we have to keep communicating and make sure that it carries on, so that we can have a spirit of adventure and fun in science, and that will underpin big breakthroughs. Thomas, it's been wonderful having you here and talking with you. I hope it will be the first of more future conversations, and I wish you every success in continuing to spread that message and emphasizing why we should all be having fun in science and doing the most exciting discovery possible. Thank you very much.
Thank you, Angus.